Today we're going to do lecture 13, looking at viruses. Viruses are acellular agents, so they are non-cellular. It also includes the group like the acellular agents include viruses, viroids, and prions. These are all going to have in common that they lack most of the characteristics of life, and they are obligate intracellular parasites. When we look at the characteristics of viruses, they're going to have one or several pieces of DNA or RNA, but not both. They don't have a cytoplasmic membrane. They may have an envelope, but they will lack a cytoplasm, except with one exception, they lack organelles. So they don't have their own metabolic activity. They have to take control of the host metabolic machinery. They have to use the host to make new copies of themselves and reproduce. The virion is the extracellular state of the virus. You're going to have a capsid or protein coat around a nucleic acid core called the nucleocapsid. The outer layer will provide protection and recognition sites. This is going to be involved in penetrating the cell, the host cell. The envelope typically is going to fuse with the cell membrane of the host cell. Once inside, they'll go into their intracellular state. The capsid is removed and they'll exist as a nucleic acid. The viral genetic material can be double-stranded or single-stranded, DNA or RNA, linear or several molecules. They have smaller genomes than the cells. The phage MS2 only has 3 genes. E. coli has over 4,000 genes. So your viral hosts, they're going to be very specific. So the virus is going to have an affinity for surface proteins or glycoproteins, for the complementary proteins or glycoproteins to what they have. It is specific not only to the host, but sometimes even for the type of cell in that host. The host range is going to be the spectrum of hosts that virus can infect. Your bacteriophages or phages are viruses that cause bacteria. Viral sizes are small. They're 10 nanometers to 300 nanometers, about the size of the smallest bacteria. The virion is your complete, fully developed infectious particle that's going to be composed of the nucleic acid and the protein coat outside of the host cell. Your capsid is going to be a protein coat that's used for attachment and protection. When we look at its morphology or shape here, the capsomeres are the proteinaceous subunits that make up the capsids. You can have a single or multiple types of protein involved with this. The viral shapes can vary. The helical ones, the capsomeres are bound in a spiral to form a tube around a nucleic acid. They'll be more of a rod-shaped. We can have polyhedrals that are roughly spherical. They're going to look similar to a geodesic dome. The icosahedron with 20 sides is most common. And then others will have more complex shapes, some with an icosahedral head with a genome and a tail fibers that will look like a spaceship. The viral envelope is going to be the membrane around the capsid. This is acquired from the host during replication or release. It's going to be a phospholipid bilayer with proteins and some viral encoded glycoproteins that will appear as spikes on the outside. The envelope proteins and glycoproteins are often involved in host recognition. Some virions are enveloped and others are non-enveloped or naked. The naked viruses are more stable outside the host, but they're more susceptible to attack once they're inside the host. When we look at the classification of viruses, this has taken a while for this to happen. They're categorized by the type of nucleic acid, their envelope, their shape, and their size. The family is the highest taxonomic group. There's several viral orders, no kingdoms, divisions, or classes that are established. So when taxonomy was being developed, they didn't even know about viruses. So with the names that they've come up with, the suffix virus is used for genus names. The family names end in viridae. Order names end in ALES. So for example, the family Herpes viridae, you have the genus simplex virus, and the example would be human herpes 2. When we look at viral replication, they cannot replicate themselves. They have to depend on the host. This is a website here that's got some more information on viruses if you're looking for a little more. With lytic viral replication, you've got your 5 main stages. You're going to have attachment of the virion to the host. entry of the virion and its genome into the host. It will then synthesize nucleic acids and proteins with the host cell machinery. It will then assemble the virions within the host, and they are released from the host. The example we use is the T4 phage that infects E. coli. It's going to use the lytic replication cycle. So attachment occurs by random collision. Since the virion is non-modal, it has to just bump into the host. Once it makes contact, the T4 is going to release lysosome to weaken the cell wall, and the tail sheath forces a hollow tube and will inject the genome, with the capsid being abandoned on the outside. The viral enzymes will enter and then degrade the host DNA, and then it will begin synthesizing viruses. The capsomeres accumulate to form the capsids, and then they will attach the head and tail fibers and insert the genome after it's assembled. This process is spontaneous to assemble the viral particles. The lysosome continues to disintegrate the cell wall, and then new virions are released. In T4 lytic replication, it takes about 25 minutes, and it will produce 100 to 200 virions for each cell that is lysed. There's a video animation of this that you can watch. Lysogeny is a modified replication cycle. Here, you're going to have the infected host cell grow and reproduce normally for many generations before they lyse. So with lysogenic replication of the lambda phage, we see this in E. coli. The viral DNA is going to enter the cell, but the host DNA is not necessarily going to be destroyed. The phage genome does not immediately take control. Instead, the virus can remain inactive as a prophage. So the repressor protein keeps the prophage inactive. It makes the bacteria resistant to additional infection by other viruses of the same type. The viral DNA will get inserted into the host chromosome. All of the daughter cells of that lysogenic cell are going to be infected. So the descendants may be part of the chromosome for several generations. At some point, that prophage may become excised from that host chromosome. We call it induction. This can happen at a later time. The inductive agents typically are going to be the same physical or chemical agents that would damage DNA. It gives the virus the indication that this host may not be around for long. So then you assemble the virions, and they're finally released. So there's a video here of lysogeny if you would like to see that in action. When we look at the replication of animal viruses, it's going to involve the same basic 5 steps: attachment, entry, synthesis, assembly, and release. But there are several differences due to the envelopes that are found in some of these viruses, and animal cells lack cell walls. So when we look at attachment of animal viruses, they're going to require an exact fit of the proteins or glycoproteins. Animal viruses lack tails and tail fibers, so they will have the glycoprotein spikes or attachment molecules on the capsid or envelope. When they go in for entry and uncoating, there's a few ways this can happen. You can have direct penetration, where the naked viruses, the genome enters while the capsid remains on the surface. An example would be polio. With the enveloped virus, the entire capsid and the contents enter the cell. The viral capsid is removed by uncoating. Measles and mumps are in this category. The viral envelope, here the viral envelope and the host membrane fuse, and the envelope and glycoproteins fuse as part of the membrane. Most envelope viruses are going to enter the host after attachment to receptor molecules on the cell surface, and this will stimulate the cell to phagocytize the entire virus. So the herpes virus is going to work this way. So here's a link on some animal viruses. So when we look at the synthesis of an animal virus, this is where it has to make its DNA. So with the double-stranded DNA viruses, this is going to be similar to normal replication of DNA. You uncode it, and then you use the genome as with the viral DNA as a template for RNA. It's going to transcribe the RNA. The capsomere proteins are going to be made in the cytoplasm, and then you'll have spontaneous assembly of the virions. This would include the herpes virus and papillomaviruses. Some exceptions here are the pox viruses. They're synthesized and assembled in the cytoplasm. The nucleus is not involved. the hepatitis B virus uses an RNA intermediary instead of replication, so you go from DNA to DNA. With the single-stranded DNA viruses, this would include things like parvovirus. It's going to enter the host, and it has to produce a new strand of DNA that's complementary to the viral genome so that it can have double-stranded DNA. After it's got double-stranded DNA, it will just follow the same pattern as a double-stranded DNA virus. With the single-stranded RNA viruses, sometimes we can call these positive and negative sense single-stranded RNA. So the positive sense single-stranded RNA, these are able to use that single-stranded RNA to act directly as an mRNA. This would include things like the poliovirus. The complementary negative sense RNA will be transcribed by viral RNA polymerase, and that negative sense serves as a template for transcription of the positive sense single-stranded RNA genomes that will go in your new virions. The retroviruses, they have positive sense single-stranded RNA, but they don't use the genome as an mRNA. Instead, they use a DNA intermediary that's transcribed from the positive sense RNA with reverse transcriptase in the capsid. The reverse transcriptase is going to take that positive sense DNA and make an DNA from it. And then it will also have that positive sense RNA act as an mRNA and a genome for the new virions. So HIV is an example of a retrovirus. So we actually will use the reverse transcriptase that we've gotten from the HIV virus in biotechnology. The negative sense RNA viruses, here the negative RNA is not recognized by the ribosomes. It's going to carry an RNA-dependent RNA transcriptase in the capsid, and it will transcribe a positive sense RNA from the negative RNA genome. The transcribed positive sense RNA will be used for transcription and a template to make the negative sense RNA genomes. This would include things like the rabies and flu virus. And then we can also have the double-stranded RNA viruses. The positive sense strand serves as an mRNA for translation of the proteins, and it makes a different RNA polymerase to transcribe more double-stranded DNA. Or it's actually going to be double-stranded RNA. In each strand, or it's going to make double-stranded DNA, and each strand acts as a template. So it will be the RNA, and each strand acts as a template for transcription of the opposite strand. So the retroviruses are in this category. So double-stranded RNA, positive strand serves as the mRNA template, uses a different RNA polymerase to transcribe more of the double-stranded RNA. Each strand acts as a template for transcription of the opposite strand. So assembly and release. The DNA viruses are going to be assembled in the nucleus and then get released to the cytosol. RNA viruses will develop directly in the cytoplasm. Animal viruses are usually going to take more time than phages. Enveloped viruses will be released by budding. This allows the infected cell to remain alive and slowly continue to shed viruses in persistent infections. The naked viruses will be released by exocytosis or similar to budding, and they may cause lysis or cell death. So here's a couple of videos on microbial diversity. So with latency of animal viruses, some may remain dormant as proviruses or latent viruses for years. When this happens in animal cells, it's permanent. There is no induction like the lysogeny of bacteriophages. When culturing viruses, they have to be cultured in suitable hosts. So we have to use a media consisting of mature organisms, embryonotted eggs, or cell cultures. The Plaque Method here is a way of being able to observe the presence of bacteriophages. A bacteriophage sample is mixed with the host, and then it's going to be in melted agar. Those will be poured in a petri plate over hardened layers of growth medium. So each virus in there will infect a bacteria, and then it will release new phages in the vicinity to infect more of the bacteria. Following several of these replication cycles, you're going to see all of the bacteria in an area be destroyed, and it will create a clearing or a plaque. We can refer to those as plaque-forming units, and this can be used when trying to determine the viral concentration. It assumes each plaque corresponds to a single virus that was present in your initial suspension. So viruses do play a role in cancer. So neoplasias are uncontrolled cell division in a multicellular organism, and when they form a mass, we call these neoplastic cells a tumor. Benign tumors are ones that are going to remain in place. Generally, they're not harmful, but they can rob adjacent cells of space and nutrients. Malignant tumors will invade neighboring tissues. These are also called cancer. They are harmful. Metastasis is when the cells travel to invade other tissues, and these can produce new tumors. The role of viruses in this process, it's thought that viruses contribute to 20 to 25% of human cancers. The viruses may carry oncogenes. They can insert and promote oncogenes that were already in the host, or they may interfere with normal tumor suppression in the host. So we do have other parasitic particles out there. The viroids are extremely small single-stranded RNAs that are infectious and pathogenic in plants. They appear to cause disease by adhering to the complementary plant mRNA. We do have other viroid-like agents that are infectious pathogenic RNA particles that will lack capsids. These will affect some fungi, but not plants. So the prions, these are the proteinaceous infective particles. They're not viruses. They lack nuclei. They lack RNA or DNA genes. They're replicated by causing this PRP cytoplasmic protein to fold differently. It only will misfold if methionine is in the 129th amino acid position. Only about 40% of humans have methionine in this place. So the humans that have this are susceptible to prion disease. If you don't, you're not susceptible to prion disease. Infectious prions cause disease following the ingestion of infected tissue. You can also get them by having a transplant of infected tissue or contacting infected tissue in a mucous membrane or skin abrasions. We do use extreme precautions to try and prevent prions just because we don't have any treatment for them. So prions are associated with the bovine spongiform encephalitis or mad cow, scrappy in sheep, kuru in humans. We actually don't see that anymore without having human cannibalism. Chronic wasting disease in deer and elk, and the variant Creutzfeldt-Jakob disease in humans. Prions are not destroyed by cooking or normal sterilization. It takes 482 degrees Celsius for 4 hours in an autoclave to destroy them, or 132 degrees Celsius in concentrated sodium hydroxide for an hour. 10% solution of some phenolics may destroy them, and the EU has recently approved the use of prion enzyme to destroy some prions.